Hello everyone. So this is the second part of our chapter, which is cell cycle and cell division. So in the last class we have discussed about the three further phases of our interface, which were G1 phase, gap1 phase, S phase, which is the synthesis phase, and G2 phase, which is the gap2 phase. So let's now discuss what are these phases and what happens in these phases. Okay. So first we have the G1 phase. G1 phase, it is the interval between the mitosis and the initiation of DNA replication. So it is the interval when between the mitosis and the DNA replication. Like here it will start the DNA replication, okay, and here it is about here we have the mitosis, okay, we have the mitosis, here we have the DNA replication. So the time between this is what is our gap1 phase. Okay, so what happens in the gap1 phase? In this phase the cell is metabolically active and continuously grows. I told you that growth is a continuous process, so growth will continue in every stage. Okay, so what will happen in this? Also we have like the cell growth will continue. Okay, cell growth will continue, and the cell is metabolically active also. Cell is metabolically active and cell growth will continue, but it will not replicate its DNA here it will not replicate its DNA. Okay, so what happens in the G1 phase? Nothing will happen. Just we have the cell which is metabolically active. Cell is metabolically active, and we know that the growth will continue. Growth will continue. Okay, and next, but there is no DNA replication. There is no DNA replication. Okay. Next, we have the S phase. S phase is also called the synthesis phase. So, it is the period during which the DNA is synthesized. There will be some synthesis. So, what will synthesize here? The DNA will synthesize, or we can say the replication will take place. So, in the S phase, DNA replication, DNA replication will take place, or DNA synthesis will happen. Okay, next we have so what happens? The DNA replication will happen, so the amount of DNA per cell will get doubled. Like for example, if the initial amount of DNA was 2C, then after it will get increased to 4C. Okay, so there will be increase in it, so the DNA amount will get double per cell. Okay, next we have that there is no increase in the chromosome number, but there will be just DNA synthesis, DNA replication, but no increase in the DNA number. No increase in the DNA number. Okay, so next we have we have an important note about the S phase. Like in the human cell, what happens during the S phase? DNA replication will begin in the nucleus. DNA replication will begin in the nucleus, and the centriole duplicates in the cytoplasm. Centriole will duplicate in the cytoplasm. So basically, it happens that in the S phase, what happens? There is DNA replication. So, but in the animal cell, it is important to note down that during the S phase in animal cell, DNA replication will begin in the nucleus, and then the centriole will get divided in the cytoplasm. Okay, so next we have the G2 phase. G2 phase, what happens? G2 phase is the preparation phase. Okay, it is a preparation phase. Here we have to prepare. Okay, so how we'll prepare? We will prepare by synthesizing what proteins? We will prepare by synthesizing the proteins for the preparation of mitosis, while the cell growth will continue only. Okay, so what happens in G2 phase? G2 phase में क्या होगा? Proteins are synthesized. Proteins are synthesized for the preparation. Synthesized for the preparation of what? For the preparation of mitosis, and cell growth. Cell growth will continue here only. Here also cell growth will continue. Okay, so these were the three phases which we have under the interface. Now there is something important written. that some cells in the adult animals do not appear to exhibit division. Like we have some people, like some different kind of people we have also in this world, due to like diversity in the world. We have different kind of people, different thinking the people have, different perspective that people have, right? So what happens, some cells are also like different from others. So these cells doesn't want to appear to exhibit division, like the heart cells and many other cells that only divide occasionally and only divide when they have the like needed, then only they divide, otherwise they don't divide. Like some cells are needed to replace the cells that have been lost during any injury or during the cell death. If there is some cell gets replaced, that time also only they divide. Or there will be some occasion, or we can say occasionally, it means no festival. It means occasionally means not always, but at some specific times, okay? Like when it is needed, that time only they will divide. So these cells which do not divide, they will exit the G1 phase and enters an inactive stage which we call as quiescent stage. Quiescent stage we called, and we denoted by G naught. Okay. So what will happen? These cells which are different will kept in some different place. Okay. So what happens? The cells which do not want to divide, like they do not appear to exhibit division, or some cells which only divide occasionally or when needed, these cell will what do? This cell will exit the G1 phase. This cell will exit the G1 phase and go to quiescent stage, which we denote as G naught. Okay. So cell in this stage will remain metabolically active but no longer proliferate unless they want to do, or depending on the requirement of the organism, like whether it should do or not. So in animals, mitotic cell division is only seen in the diploid somatic cells. So there are some few exceptions that this where haploid cell divide by mitosis. For example, male honeybees. In male honeybees, this is the exception. Important to note down. Again, this plant can also show mitotic division in both the haploid and diploid cell. From okay.